Well, today I'll be explaining about mitosis. Mitosis is a fun, like it's amazing. It's something that I love to watch. It's like a movie, as many people say, and it's pretty cool. So, what did I do right here? That's not cool. I did an eye for no reason. So one of the things that mitosis goes through is the cell cycle that where the cells get bigger and bigger and where they like develop each other. Develop each yeah. So the cell cycle starts on G1 where the cell is growing. Then we go to the S. That's the synthesis. It means it's copying the DNA so it can reproduce and do a exact copy of the DNA. So after the synthesis, we go to G2. G2 means growing more and more. And some cells doesn't do that. For example, muscles, they don't copy each other. Yeah, muscle doesn't do that, and nerves doesn't do that. So they go to a phase they call G0. The G0, it's right here. So you do anything like this, and a G, and a 0. Kind of easy. So if you're not dividing, you go to G0. That's not dividing the nucleus. But if you are doing that, then you go to the next phase, that's mitosis, that's the end part of the cell cycle, that we're gonna copy the nuclei and do the exact copy. Yeah, most of the people they count as a clock for the cells, where they spend their most of their time doing interface. Yeah, most of the people they count as a clock for the cells, where they spend their most of their time doing interface. And why they spend their time in the interface, where they grow and it got bigger and bigger. That's where they spend most of their time. And after that phase, they create 2 different cells, and each one of them go to do their different cell cycle. Yeah, I totally forgot to draw the interface for you. But the interface, I just saw somewhere, they share it outside. So right here, so we put E, and this outside right here, do this. Until we start the metaphase right now again. And do one more M right here. Well, now we're gonna explain the phases of mitosis. So let me get a new piece of paper. The first phase we call interphase. The interphase is where everything starts. But before the interphase, the cell used to look like this. Just that, nuclei, and 1 centraloid. But after the interphase, it goes to this. The cell changed to have 2 centraloids, and he does a copy of the DNA. So the 2 centraloids, and the middle right here, nuclei, they already did the DNA. So let's put the 2 dots and explain. So right here, boom, boom. DNA is copy. And the centraloids, replicated. As we move next to the next phase, we call prophase or metaphase. And right here, I had to do something there. I forgot to do it. It was this. Yeah. The DNA looks really messy at the beginning of the interface. But now going to prophase, where everything will change too. I guess all of them change. On prophase, the centrosomes will and the microtubules will start connecting each other, And there, let me show you how they have. I'll be doing with black, the microtubules. They look like this. Something that we see too inside the nucleus, the chromosomes start condensing each other. Instead of forming all this little scrubby right here, they start forming to a chromosome. So they're gonna look like little weird chromosomes, not fully formed, but less confusing to see like this. It will be more understandable to see it. You'll see that they're condensing and forming to a chromosome. So the first thing that happens right here, it's chromosome, condense, and mitotic, the mitotic spindle form, it's something that will help organize the division of the nuclei. And now we go to prometaphase. Prometaphase. So in prometaphase, what happened is, 1 second. That's supposed to be an E, okay? The first thing we can see that I didn't draw yet is the nuclear envelope fragments. So what that means is, this little bubble right here, the nuclei, it breaks, it fragments. So in the centrosomes, they were gonna start connecting to the kinetochore. The kinetochore I didn't show was the kinetochore, but this is a chromosome. A fully grown chromosome look like this. And the kinetochore is right here in the middle. So, on the kinetochore. Yeah, kinetochore. And the centrosome will connect right here. And we'll do this. Let me draw what happens with the fragments. So let's do a little black dot as a fragment. And just let's use a small Sharpie to show. And they're all being pulled apart. And get a good pen that can show that. Using the black pen again. To show what happens in this phase. And let me write. So the nuclear envelope fragments and the microtubes, sorry. They connect to the kinetochore. So the microtubes are the black part, so that's why I separated instead of doing yellow, because it's hard to see it and I don't have a Sharpie that's yellow and it's small enough to show you. They connect to the chromosomes right here, as I said, and the kinetochore, that's the middle part of the chromosome. So after prometaphase, we go to metaphase. Metaphase, it's where all the chromosomes, they align and do a really straight line. As one of my YouTubers that I like to listen, they said that was one of the most straight lines in nature. So the metaphase, don't confuse that with Facebook. Ha ha ha, jokes. Sorry for jokes. I think it's Meta, something on Facebook. I don't know. I don't care. But yeah, so the centrosomes, they're putting all the chromosomes in the middle, so they do one segment, so they will look like this. So let me do this, boom, boom. And what's going on right here we call the metaphase plate. So after metaphase, we go to anaphase. Anaphase, we have to pay attention to the chromatids, because the chromatids will break apart, and I'll represent that to you so you can see it. What happens? So in metaphase, let's do like a little egg, more oval. We got the centrosomes right here again, obviously, because the centrosomes is the most important part that helps you understand what's going on. They'll be pulling apart the chromosomes, and they will look like this. As you see, you can see that they're getting break apart, and so let's keep a note saying what happens on anaphase. For mitosis, it's chromatids. Separate. So after anaphase, we go to telophase or cytokinesis, and we write that like this. So what's going on the telophase? It's that the cells are dividing and they're creating a new cell. So it looks like this. It looks like a number 8 that's breaking apart. And right here you'll see a cleavage that means that the centrosomes are pulling apart and helping them break apart. And with the chromosomes or chromatids, they're gonna start creating a new nuclei. The daughter cells will start creating a new nuclei. So this is the daughter cells creating a new nucleus. And we have, again, the centrosomes. Centrosomes are the one that are assisting a lot to help them move apart. You will see the microtubes right here helping them move apart. And yeah, that's metaphase. And the cells are gonna go in the same process again and again and again, and they will go to the same thing again. Yeah, thank you for listening, and hope you like it. And sorry for my stupid jokes that I think this is making no sense. But thank you for watching.